Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with ShahTareen Swati. And I'm here to discuss the most important organelle of the cell, and that is endoplasmic reticulum. As you can see here on the screen, the endoplasmic reticulum, which is the most important organelle of the eukaryotic cell, but this organelle is absent in prokaryotic cell. But before starting my lecture, I have a very humble request to all of you to do subscribe my channel, like my videos and share my video. If you are sharing my video, you are sharing the knowledge. So keep watching, keep sharing knowledge with other. Now I am going to start my lecture. I will discuss about the history, structure, type and function of the endoplasmic reticulum. Okay, first of all, what is endoplasmic reticulum? I will show you here. As you can see, this is the endoplasmic reticulum structure. You can see here, this endoplasmic reticulum, the word is derived. If I talk about the word derivation, the word endo means inside, plasmic means cytoplasm, and reticulum means network of channels or canals. So you can see here the endoplasmic reticulum present in the cytoplasm in the form of what? Network of channels or canals. So endoplasmic reticulum basically consist of many other parts, like most important, it's one single part is known as the cisternae, which I will discuss later on, and tubules and vesicles are all the part of the endoplasmic reticulum. So the endoplasmic reticulum is a membrane bounded organelle which includes smooth tubules and rough sheets, while the ER is defined as an interconnected network with a continuous membrane. The different structures that make up the ER perform very diverse and specialized function within the cell. So, as the name indicated, that this is the network of channels or canals. So, its basic function is transport of the substance within the cell and outside of the cell, but its other most important function I will discuss later on. Okay, so first of all, its discovery. The endoplasmic reticulum was observed with the light microscope by the Garner in 1897. So Garner in 1897, who coined the term ergastroplasm. First, its name was ergastroplasm. With electron microscopy, the lazy membranes of the endoplasmic reticulum were first seen in 1945 by Cat R. Porter and Albert and Ernest. Later, the word reticulum, which means network of channels, was applied by Porter in 1953 to describe this fabric of membranes. Is it clear? So this is little about the history of the endoplasmic reticulum. Okay, now I'm going to discuss the structure of the endoplasmic reticulum. So hold on your notebook, pen and pencil and draw the structure of the endoplasmic reticulum. ER is composed of three kinds of structures. This endoplasmic reticulum consists of how many structures? Three kinds of structures. You can see here, these are the cisternaes. These membrane bounded channels or canals which are parallel to each other are known as cisternae. And you can see the endoplasmic reticulum start from the nuclear membrane and up to the cell membrane. So it makes the connection. It moves the substance from the nucleus to the what? To the cell membrane. So you can see here, there are small granules-like structure present on the surface of the endoplasmic reticulum. And on the basis of the presence of these ribosomes, these granules on the surface of the endoplasmic reticulum divided into two forms or two types. And you can see here, these channels, these canals, these cisternaes are without granules or the ribosomes. So this is the smooth type endoplasmic reticulum, and this is the rough type. And these channels or canals are known as cisternae. The other, you can see here the rounded spherical structures, these are known as the vesicles. These are also pinched off from the cisternaes, endoplasmic reticulum cisternaes. And the third one is the tubule. So what is the difference between the tubule and the cisternae? The difference I will show you later on. The tubules are branched and cisternae are unbranched. And you can see there is a space between these channels and canal. This is known as the lumen. This is known as the lumen of the cisternae or endoplasmic reticulum. And inside this lumen, you can see this is the lumen. These are the cisternaes which are covered with the ribosomes, and these are lacking the ribosomes. So this is smooth type, this is the rough type. Is it clear? Okay. Now I'm moving towards the cisternae. These are long, flattened, sac-like, unbranched tubules. As you can see here in the diagram, these are what? Unbranched. Okay. Now, these are often arranged in a parallel rows forming bundles as in synthetically active cells of the pancreas and liver and many other cells. Each eukaryotic cell contain what? The endoplasmic reticulum. So each cisternae is 40 to 50 micron in thickness, having ribosome on its surface, as I have shown you in the figure. In mammalian liver cell, parallel cisternae of more or less uniform size are present during metabolic activity, whereas in pancreatic cells exhibit a slightly different arrangement of cisternae. So this arrangement and the size of the cisternaes are different in different organs, or in different tissues, or in different cells. Is it clear? So its its shape depends upon its size depends on the structure as well as the function of the cells in which they are present. Is it clear? Okay. Now, cisternae show different arrangement. As in mammalian liver cell consists of the parallel cisternae, while pancreas has different systems. In striated muscles, this endoplasmic reticulum is arranged in a network of tubules, and that is called the sarcoplasmic reticulum. Are you familiar with the sarcoplasmic reticulum? No, if you are not familiar, so I am telling you here the sarcoplasmic reticulum is the name of the endoplasmic reticulum, but in the muscle cells. Is it clear? Okay. Now I'm moving towards the figure, and you can see here again, I'm showing again and again the cisternaes and the vesicles and the tubules in order to make your concept clear. Is it clear now? Okay. Now, vesicles. These are usually rounded or ovate structure, ranging in diameter from 25 to 500 micrometer. They are ovate structure and rounded. The unit membrane is of the same structure as plasma membrane. Is it clear? Vesicles often occur isolated in the cytoplasm. They are found in abundance in pancreatic cells, because pancreatic cells secrete the pancreatic juice as well as many other enzymes and lots of things. You can see here, I'm here clearing one point upon you people, that the endoplasmic reticulum, when pinch off and form the small vesicles, these vesicles move towards the cytoplasm and help in the formation of the Golgi bodies. Must keep it in your mind. The Golgi bodies are formed because of the fusion of the small vesicles which are produced by the endoplasmic reticulum. And these form Golgi bodies, and these Golgi bodies, each part is also known as cisterna, which I am going to discuss in my next lecture, inshallah. Okay. Now you can see here, these are the vesicles, these are the cisternae, and these are the These are the tubules which I am going to discuss. Is it clear? So cisternaes are without branch, they are unbranched, and vesicles are rounded or spherical, and the tubules are branched. Okay. Now vesicles are surrounded by coating protein. This is very important information for you people, that these vesicles are coated with a coating protein which are called COP1 and the COP2. Means CO for coating, P for protein, 1 and 2. So COP2 target vesicles to the Golgi apparatus. Must keep it in your mind. And COP1 marks them to be brought back to the rough endoplasmic reticulum. COP1 targets the vesicle to the Golgi bodies. Is it clear? And COP2 vesicles target the vesicle to the Golgi apparatus, and COP1 mark them to be brought back to the rough endoplasmic reticulum. Must keep it in your mind. The rough endoplasmic reticulum work in concert with the Golgi apparatus to target new protein to their proper destination, because I have shown you that the Golgi complex is formed by the fusion of the vesicle which are produced by the endoplasmic reticulum. The second method of transport out of the endoplasmic reticulum involve areas called membrane contact sites, where the membranes of the endoplasmic reticulum and other organelles are held closely together, allowing the transfer of the lipids and other small molecules. Is it clear? Okay. Now I'm showing you here COP1 and COP2 proteins. You can see here the coated protein, the vesicles. You can see here clearly blue color, and other are represented by the green color. And you can also see here the clathrin, which I am not discussing here. So these are also the coated vesicles, but here I am just showing you the COP1 and the COP2. The COP2 function is to target towards the Golgi apparatus, and COP1 back to the endoplasmic reticulum, as you can see here in the figure. Is it clear now? Okay. Now I'm discussing the tubules. These are of diverse shapes and irregularly branched, having diameter of 50 to 100 micrometers. These are found in the cells that are active in the synthesis of steroid compounds like cholesterol and glycerides, etc. These are also found in the pigmented epithelium cells of the retina. Must keep it in your mind. And the function in the retinal pigmented epithelium is the vitamin A synthesis. Is it clear? Okay. Very interesting. And now ultrastructure of the endoplasmic reticulum. All the three structures of the endoplasmic reticulum are bounded by a thin membrane of 50 to 60 angstrom thick, like plasma membrane, nucleus, etc. All are membrane bounded organelles, and their membrane have the same lipid bilayer. Its membrane is also formed of the three layers. Outer and the inner dense layer are composed of the protein molecule, and the two middle layer and transparent layers are of phospholipids. Is it clear? Now endoplasmic reticulum membrane is continuous with the plasma membrane on one side and the nuclear membrane on the other side, and also with the membranes of the Golgi complex, because the Golgi complex is produced by the fusion of the vesicles which are produced by the pinch of oil from the cisternae of the endoplasmic reticulum. So the lumen of the endoplasmic reticulum act as a passage for secretory products, and Platt has observed secretory granules in it. Is it clear? Okay. I hope so the lecture will be clear upon you people. Inshallah, in my next lecture I will discuss about the types of endoplasmic reticulum and the function. Till then, Allah Hafiz.